Good afternoon, everyone. A warm welcome to members of the department, other members of the university, and some of our alumni that have come along today. This afternoon's lectures, an important facet of the Department of Genetics life, which was established some 8 years ago under Anne Ferguson-Smith's leadership. So I want to welcome you to the fourth biennial Saunders Genetics lecture. So we set up these lectures to celebrate the life and contribution to genetics and heredity that Edith Saunders made at the turn of the 20th century. Edith was born in Birmingham in 1865 and obtained a scholarship and entered in Newnham College in 1984, obtaining a first-class honours in the natural sciences tripos in 1888. Obviously, being a woman, she could only get a certificate, and it wasn't until 4 years after her death that women in Cambridge were finally awarded degrees. But nevertheless, while at Newnham, she met William Bateson's two younger sisters and had an interest in natural sciences, particularly in botany. The Balfour Laboratory for Women was established at Newnham to facilitate allowing women to do research in Cambridge. At that time, although they could go to lectures and take the examinations, they were under no circumstances permitted to attend practical classes. After her graduation, she became a demonstrator in the Balfour Laboratory, later rising to become head demonstrator at the Balfour. She was a lecturer at Newnham College, and for a period of time she acted as a demonstrator in the Department of Botany when botany was an extremely popular and one of the largest departments in the biological sciences. From our very earliest days in Cambridge, she was interested in plant breeding. Of course, the term genetics had not been coined there, but she very soon began to work extensively with William Bateson, who was a very prominent supporter of the development of Mendelism at the turn of the century, and really during the later part of the 19th century and right until William Bateson's death in 1926, they worked very extensively together, including the time he spent at the John Innes Horticultural Institute, the predecessor to the current John Innes Centre in Norwich. Importantly, however, Edith carried out a multitude of independent research, and her early work on mustard plants was very influential on Bateson in supporting his idea of against gradualism in terms of phenotypic mixing. And she also wrote standard texts on floral morphology. But particularly important, from 1902 to 1908, Bateson and Saunders wrote 4 very important reports to the Royal Society outlining the basis of Mendelism, and these were widely regarded as the start of genetics. Bateson always regarded Becky Saunders as a collaborator. There was no question of Saunders working for Bateson. Our impact was unbelievable. She was the first woman elected as a fellow of the Linnean Society, and later became the first president of the Linnean Society. A fellow of the Royal Horticultural Society in 1925, and indeed received its Banksian Medal in 1906 at the Third International Congress of Genetics. She was a life member of the British Association for the Advancement of Science, and as these plaques, which we'll get to at the end, indicate she, along with Bateson, was founder and former president of the, at the time, Genetical Society, which we now know as the Genetics Society. As I said, sadly, Saunders was never a member of the university. Women didn't get degrees. They couldn't get jobs that men could do. You know? But her contributions to genetics were immense. JBS Haldane, a senior geneticist at the John Innes and noted socialist firebrand, noted in her obituary in Nature. Actually, he was outraged about the obituary that was published in Nature about it, so he wrote a second one and noted that Saunders' work was of the first importance in genetics, and also suggested that Edith Saunders was the mother of British plant genetics. So it's very fitting that this year's Saunders lecture is being delivered by another giant in the field of plant genetics, and also given the connection to Bateson and the John Innes Centre. Professor Dame Caroline Dean is a Royal Society research professor at the John Innes Centre in Norwich, which she joined as a group leader after her undergrad and PhD at York, and a postdoctoral stint at the Advanced Genetic Sciences company in the US. Caroline's a Fellow of the Royal Society, a foreign member of the US National Academy of Sciences, the German National Academy of Sciences, and a member of the European Molecular Biology Organisation. She is a recipient of many distinguished awards, including the Royal Society Darwin and Royal Medals, the Genetic Society Medal, the Febs Embo Women in Science Award, the Leoro UNESCO for Women in Science Award, the Wolf Prize in Agriculture, and last year she was awarded both the Barbara McClintock Prize for Plant Genetics and the Genetic Society Mendel Medal. Caroline has made outstanding contributions to understanding developmental timing in plants, with a particular focus on how plants are able to remember and respond to environmental cues. And today she will deliver the Saunders lecture entitled Co-transcriptional and Memory Mechanisms Registering Winter. So, Caroline, thank you very much for...
Thank you, Steve. It's a real pleasure to be here. You know, being at the John Innes Centre, we hear about Bateson a lot. He was the first director. But I have to say I hadn't heard about Edith Saunders. So it was a treat to read up on what she's done. And I think it's wonderful to have these seminar series to acknowledge her contribution and so that we remember the name. And, you know, when you read the literature, it's what they've done for the field of genetics and also for the field of plant biology. They're hard acts to follow. But hopefully in my seminar today, you'll see this theme of genetics and how important it is and the use of plants to discover fundamental discoveries. So the talk today is about the environment. And, you know, it's a topic of the moment for all of us. We're worried about, in our own bodies, the exposure to all sorts of different things. We worry about how much of that is transmitted to our offspring. There's lots of work on that, lots of concerns. There's clearly also a lot of work and worry about what's happening to our natural plants and our crops as climate changes and the yields start to fail. Are we going to feed ourselves? It's a big topic. So an important piece of work that's happening in a lot of fields is really this trying to understand how the environment affects this genotype-phenotype relationship. And you know, most people, when you look at how they do these experiments, and there's a lot going on, start off with the presumption that they need to understand, define the environmental stressor. They'll then figure out the signal transduction pathway and work out the change gene expression that occurs in response to that. And I went into the literature and found a fairly recent paper, and this sort of shows an example of what might be happening. People will look at various different stresses, whether it be very complex ones or simpler ones, and then use the power of omics as it is now to describe the changes in response to a time course of exposure to this signal, and then look at the gene expression to understand the change in phenotype. And actually, a lot of work in environmental exposure research follows this logic. And what I hope to persuade you of today is that actually this logic sort of hasn't helped in really understanding how the organism is understanding its environment. So we'll see if I manage to persuade you of that by the end. Now, a lot of work does go on in humans, in mammalian cells particularly, now that they're easy accessible for research. But I would argue, perhaps as Edith Saunders would argue, that actually a better system to look at the environmental exposure and how that affects the organism is to use plants. Because plants can't run around, they have to use the environment for the benefit as well as sort of protecting themselves from it. They use short-term signals to change their metabolism. They use medium-term signals for all sorts of developmental consequences. And they use these long-term signals as well for timing, as Steve mentioned. And what got me into this was actually, as a postdoc in California, I sort of missed the seasons. And so I went and bought some tulip bulbs in a shop, and the man said, put them in the fridge for 6 weeks. And that one comment sort of got me into thinking about, how does an organism understand it's had this long-term exposure? Now, people have looked at this for a long time. As I'll show you in a minute, it's very important for growing our crops. But if you think about what the plants are actually monitoring as they're going through winter exposure, what you see on the right here is a graph with temperature sensors next door to some plants that we happen to be growing in a field in Sweden. And through the year, we took measurements, and what was striking was just how extensively noisy this real natural temperature was. And somehow, when you do experiments in the lab and you give cold or warm, you give periods, days of a certain constant temperature. But the plants aren't seeing that. This is somehow, they're trying to understand when winter is happening through this temperature exposure. Now, if you look at this, you think, well, maybe this is when plants are monitoring winter, this kind of cold period here between November and March. And this was when there was snow on the ground, so that was why the temperatures were fluctuating less strongly. But in fact, through transplant experiments that we've done, if you take plants out, they have fully registered winter by this November period. So this raised two questions, really. How on earth do they really understand this noisy temperature? How do they read that as to this is, winter is progressing? And obviously, too, it's not just what the temperature is that day, but they have to remember what the temperature was last week, the week before that. They have to somehow look at the memory, have a memory of prior exposure. And as we stared at this graph, actually it made us really think about these more general questions about exposure. Because most signals, if you think about going back to the human situation, chemical exposure, whether you're smoking occasionally or how much wine you have at night, they're sort of not one exposure and then you look at the effect. It's the long-term, often low-level exposures. So what I want you to do is, I'm going to split my talk into answering these two questions, basically. How does the organism, let's keep it general, dissect a signal or extract a signal from this noisy environment? And secondly, how is that registered so that you can remember prior exposure and sort of have a cumulative effect? And I want you to think about this in terms of, actually, if you think about other natural signals for plants, nutrient, other temperature aspects, complex biotic interactions are very complex signals perceived over long time scales, but also stress, diet, aging in mammals. So perhaps instead of thinking of this one sensor and this signal transduction, we should be thinking about it potentially, as I have here, in these two particular questions. Okay, so the seminar will focus on those two questions. Now, it doesn't take long when you then start looking about the exposure of winter, which is what we've been focusing on, and how plants can do that, to take you to this process called vernalization. So this is very important for many natural plants, but hugely important in our crops, breeders have used these extensively to generate winter-type and spring-type varieties of many of our major crops, winter wheat, winter rye, all of the brassicas. And so that the idea is if you need a vernalization requirement, the plants germinate early in autumn and then overwinter vegetatively, and then only after winter will they flower in the spring. And this, of course, then aligns, if you need a period of cold before you flower, this aligns flowering, a very sensitive phase of plant development, with the more favorable conditions of spring. So I show broccoli here. This is very important for, the vernalization requirement is very important for breeding a lot of broccolis. And the physiology of vernalization was well characterized in that it was a rather slow process. This is weeks and months of cold exposure. It's quantitative in that if you give more weeks of cold, you get a progressive acceleration of flowering. It was epigenetically stable through cutting. So if you took a cutting off a plant, it would kind of remember how much cold it had had. But it was always reset every time the plant went through meiosis. So these were the features of vernalization when we started the work that we were interested in understanding. And actually, it's so important, vernalization, for many of the crops, that there are many commercial models now which have taken the day-degree formula as you measure the temperature over a period of time, that come up with thermal time calculations in a sort of how much cold over periods of time, assuming some kind of gradual accumulation of something or some gradual removal of something, and that's what the plant is monitoring for, this prolonged accumulation, exposure to cold. So this was the idea at the beginning of our work with the classic signal transduction gene expression sort of mode. Now, before I launch into vernalization, which I will do, I just want to put this into a slightly bigger picture, because when I gave this seminar, people, especially the gardeners in the audience, say, but Caroline, many plant species don't seem to want cold. They need particular day lengths, or they need certain other features. If you go to the desert, they need water to flower, and if you go to equatorial regions, they need cool temperatures at night. So how does your work relate to flowering more generally? And this was actually the beginnings of the Arabidopsis explosion in the UK here, which sort of happened in the early 90s, was that the realization that you could take Arabidopsis and do the genetics, and single mutations would influence the behavior, the physiological behavior of this one plant species. So what do I mean by that? Well, Arabidopsis, if any of you have seen it, you'll probably imagine it would be a plant that looks like this. It's the sort of Drosophila of the plant world, as people used to call it. It's for rapid genetics. It's for molecular genetic approaches to cloning genes. So everybody wanted rapid cycling types. But not all Arabidopsis are rapid cycling. They actually, most of the accessions, if you went certainly from around here, collected some seed and put them in your nice warm long day chamber, would look like this. They would need cold, sort of 6 weeks or so of cold to look like this. So there is this requirement for vernalization in many accessions. They're winter annuals, as you call them, rather than rapid cyclers. But they're also very sensitive to long day photoperiods. But you could take mutants in this, starting from rapid cycling types, and change all sorts of behaviors to make Arabidopsis look like other plant species. And that very quickly clarified flowering time regulation for all plants, in that actually what it looked like was, rather than different molecular mechanisms underpinning these different physiologies, what we had was a series of genes, which we call christened floral pathway integrators, and they were regulated by many different pathways. And the fact there were common targets meant that different plant species would then use different pathways differentially, dependent on where they were growing. So if it was an advantage to overwinter, they would then have selected for the pathways that confer vernalization. For other types of Arabidopsis, it would perhaps be a photoperiod selection that would have happened. But this is why we had the grandiose title, Arabidopsis, the Rosetta Stone of flowering, because the genetic analysis quickly, if you like, elaborated what was going on for flowering much more generally. This has been a very useful framework for interpreting flowering regulation in many plants. The other surprise was when we looked at the natural variation in Arabidopsis. Now, the red here shows vaguely where all of the accessions have been collected. I think it's reflects where researchers have looked. So we haven't been a sort of comprehensive analysis of where Arabidopsis thaliana grows, but it's pretty extensive range. We've been working at the very northern latitudes, and I did some great trips up to the North Swedish part of the world, collecting Arabidopsis, but it can also grow as far down as these Cape Verde Islands near the equator. So very different geographic climates in this geographical range. Now, when we've then gone in and looked at the genes underpinning that natural variation, and of course the flowering varies very much in these different accessions, we find that if we look at the flowering network as defined by our mutagenesis of our rapid cycling type, which I'm not going to get into, it looks messy, but 3 genes have come out as the major nodes for where the natural variation has occurred. And I think this is quite interesting. And it so turned out that the vernalization pathway was a key part of that. So what you're going to hear about tonight is this gene FLC. And I'm only going to talk about one gene because all our experiments, whether we were asking, does the Arabidopsis need vernalization or not, how much coal does it need to fully vernalize, and what's the natural variation behind adaptation to such different climates, we kept coming back to this one target. So what's interesting, it has become the evolutionary node in Arabidopsis thaliana, but that is also true all the way through the related brassicas, the broccolis, the cauliflowers, the whatever. So this is a very interesting node, and I think the interesting question, which we still don't understand, most of these genes are not flowering specific. So how has evolution selected some genes and not others for this variation and underpinning this adaptation? Okay, so let's get back to vernalization. So we're going to study one gene, FLC, flowering locus C, named many years ago. It encodes a repressor of flowering. It's a transcriptional repressor, which keeps the genes off for that floral pathway integrator set of genes. So if you express the protein, you don't flower. And that's all you really need to remember. If you look at Arabidopsis vernalization, it looks very like the brassicas. We start now with a winter type, which has very high levels of FLC upon germination. This means it's making it, flowering is blocked. And this is what it looks like. With increasing weeks of cold, you get this progressive acceleration of flowering, just like in the brassicas or the wheat situation. And that fits with the slow silencing, transcriptional silencing of FLC during this long period of cold. And what I put on here is the temperature fluctuations to remind you that somehow this nice graded reduction in FLC expression somehow is being driven by these enormously fluctuating temperatures. Then what happens as the plants come back into spring and the temperatures get warmer, FLC stays epigenetically silenced. And actually it's one of those epigenetic mechanisms that people would go back to, because you can take a cutting of this plant, tiny little cutting, regenerate a whole new plant. So many, many, many cell divisions later, and FLC will still be silenced. So epigenetically very stable. Whatever happens in the cold, it's epigenetically switched off stably. But then every time you have the seed formed, as the plants flower and the seed forms, the gene is switched back on again, because of course every generation needs to go through that requirement for cold. So what we have is really simply the gene is on, epigenetically silenced in the cold, maintained in this off state while all of the other things required for flowering, long day photoperiods, warm, sugars, all sorts of things, induce the flowering, and then it's switched back on again. So epigenetically off, switched back on. Now, when we first started, of course, we wanted the power of genetics. We didn't really know what was involved. So we just did a classic forward genetic screen, finding mutations that could no longer respond to the cold. We start with late flowers. We do EMS mutagenesis. Most of the time, after vernalization, then the plants flower early. So we take these rare ones that flower late. And these are the basis of our vern mutant phenotype, vernalization mutations. And they have very high levels of FLC because they haven't been silenced. This took us into the world of chromatin, polycomb repressive complex II chromatin, in fact, which is a very conserved chromatin silencing mechanism. And so what's been interesting, I would say, over the last 5 years, my lab has sort of ended up being very, very heavily into chromatin. And so most of the time I actually don't talk to plant biologists because this chromatin silencing mechanism is just as important in mammals and Drosophila as it is in plants. It was discovered in flies. I'm sure people here worked on it over many years. Because actually the flies that they were selecting were flies that had the wrong body plan. So here shows how various things were formed in the wrong place. Here was an additional pair of wings, and this is somewhere you will know better than me. The Holters were wrong. Anyway, through a lot of analysis, what was clear was in the flies, these genes were transcriptionally set, and then they were epigenetically maintained in that state by this polycomb repressive complex 2 machinery. So it associates with elements in the gene which the polycombs associate with, and it confers this epigenetic memory, just like I said in the plants, that even in the absence of the regulators, then the genes stay in that epigenetically maintained state, and it's therefore involved in long-term silencing. And it's these days, everyone, when you say polycomb, immediately thinks of a very specific modification to one of the histones, histone 3 lysine K27 trimethylation. And I'm not going to get much into all of those. I'm just going to show those color coding as a red blob for the histone tail in a sort of traffic-like system. So if the gene is on, it's green, and these are the modifications associated with the active state, red off. So traffic-like system, when you see, that's what that will mean. Oops. So of course, we hit, with our mutants, cloning the genes, we hit the polycomb. So the first thing to look at was this accumulation of this red mark, K27 trimethylation, at the locus itself, to address why was it slow and why was it quantitative. Now, if you look now with chromatin IP and track along the gene, and this is the structure of the gene on the bottom here, this is where the transcription starts, these are all exon sequences, you can see that as the plants are exposed to more and more weeks of cold, you get more and more K27 trimethylation. So that was very satisfying. We were getting an accumulation of the silencing modification over the same kind of time frame as we were getting acceleration of the flowering. And then someone said to me, and this is work, actually, I've extended collaboration with Martin Howard, who asks always the great question as a computational biologist, but Caroline, is this accumulation at every gene a little bit at every gene, or is this accumulation, it accumulates or not in an on-off switch, and it's just the proportion of genes that have now accumulated this mark? And of course, when you do chip experiments, you can't distinguish those two. So we went in and made fusions where we could actually ask that question. So we made a fusion with Venus to the FLC. We're imaging roots here because A, they're easy to image roots, but also vernalization, the plant, the whole plant temperature senses the temperature. Okay, so you can image any bit and you'll see the same thing. And what you see on the right is the real data, and what you see on the left is a sort of schematic representation of that because it's a bit easier to look at. The cells that are dividing are at the base of the meristem here. And then what happens is that all of the cells are in what's called an anticlinal division. So this one here, this file that stays green, all come from a daughter that starts off in the meristem down here. So the beauty of this is you can track the epigenetic lineage, if you like. So in this particular situation with 8 weeks cold, most of the meristem cells switched the gene off, except this one stayed on, and all the daughters then remained on. So that's this one here that you can see. Because if you grind this up, it looks beautifully quantitative. But it's not quantitative, it's a switch. Now this mosaic type phenotype is actually average because the downstream target of FLC, this gene FT that I showed you on one of the schematics, moves around the plant. So you don't have bits of the plant flowering and bits not. It's averaged out quantitatively. The other thing that was really surprising and came out of a bit of an argument about, in the old days, histone modifications got everybody excited. But then there were groups of people who thought this was just fluff that happened on chromatin and really wasn't important at all. And one particular person in particular was Mark Potashny. And if any of you have ever been in an audience with him giving a seminar, he would then interrupt and say, ha, ha, ha. histone modifications, they're just fluff. They're all downstream of a transcriptional regulator. What's really important is the transcriptional regulation. And I remember talking to him one day and saying, well, there's sort of shades of grey here in painting. He said, do an experiment that proves to me that it's not a transcriptional cascade that you're looking at here. And so we basically did that experiment by putting in 2 fusions into the same nucleus. So we have an FLC Venus and an FLC Cherry, make the transgenic lines, and then ask, with graded amounts of cold exposure, are those switching at the same time? Because if cold was working through a transcriptional regulator affecting the chromatin, they would all be working, so whatever was remembering the memory was working in trans, it would affect both copies similarly. If it was a cis-mediated event, i.e. it's the local chromatin that's really important, then they'd switch independently. And they switched absolutely independently. This was in 2017. So here's the cherry. It's a bit hard to look at, but when you look at both together, you can see that they don't align. So this basically said that this switch, so it's an on-off switch that happens quite slowly, and it needs to be slowly because the plant needs to have gone through the whole of winter before it fully silences the repressor, is driven by local chromatin and not by a trans factor network. And, that's from a sort of epigenetic field, a very important conclusion. So what do we know about this switch? Well, I'm not going to go through all of the details, but it's clearly involving the PRC2 complex, which let's just call these various grey blobs here. The names are different in every species, which makes it very hard, but these are very conserved 4 components of the classic PRC2 complex. But the other thing that came out were 2 proteins out of the screen, which I'm going to call PRC2 accessory proteins. And certainly in the mammalian field, those have become very important now for giving specificity to where polycomb silences in the genome. And one of those, when we looked at our time course, was very slowly induced by cold. This is a protein called, a gene called Vin3. And actually the dynamics of accumulation were a little strange. And again, I attribute this to sort of working with Martin. Something that accumulates in the cold has a profile more like up and plateauing. This accumulation dynamic said something a bit interesting to him. And I'll get back to that in a minute. So what we've got is when the switch happens, this group of proteins stays associated with this very small region. Remember the chromatin IP I showed you was actually a quite localized region in the gene. And when the switch happens, that protein complex binds and then stays there, even through cell division. That's the basis of the epigenetic memory. So then the 2 questions are, well, why is it so slow? And could it be something to do with this slow Vin3 accumulation? And why is it then epigenetically stable? What keeps it there? Now, why it's so slow took us into looking at Vin3 regulation, and I put this in because it was such an unexpected result. The direct regulator of this cold-induced Vin3 turned out to be a protein which directly regulated the promoter of Vin3, but wasn't itself affected by temperature at all, because we imagined it would be. The protein wasn't affected, the translation, the RNA, nothing. It turned out to be that it was a very stable protein, and it was only expressed just in the meristems of again, we're looking at roots. So this is the GFP fusion to that protein, happens to be called NTL8. And in the cold, cell division slows down. And because cell division slows down, it is diluted less effectively. And so it accumulates. And that's what it gave us, that rather interesting accumulation pattern. So here we have long-term temperature sensing through effect on growth and not a direct effect on gene regulation. And again, I think that's probably got some fairly generic messages behind it. So that's why it was slow, because it was dependent on the growth. But also we had an interesting finding in that one experiment we did where we were looking at the fluctuating temperatures, and we were fortunate in Norwich to have chambers which we could mimic the fluctuations that we saw in the field. And we had fluctuations, so we either gave the plants a constant 8 degrees for 24 hours every day, or a fluctuating 8, but over the 24 hours would have 8, or a constant 14 or a fluctuating 14. So this is as we were trying to understand which aspect of the temperature the plants were following. So the plants got this temperature regime in the 24 hours, and they were grown then for different days, and we looked at the VIN3 levels of expression. Now, if you were around 8, fluctuating or constant made no difference. The plants saw the same temperature input. But where we looked at the fluctuating 14 versus constant 14, we saw a big difference in VIN3 accumulation. So what this said to us was that we were getting this accumulation, but somehow within the very fluctuations which gave these very high temperature peaks, and these are the afternoon peaks, which of course temperature gets higher during the day, all the VIN3 that was accumulating was turned over. So we had these different aspects to regulate this long-term accumulation. And for the first time ever, it said that thermalization relied on the absence of warmth as well as the presence of cold, which actually, when you look at the fluctuating temperatures, makes perfect sense, but had never occurred to anyone before. Okay, so the second question was, well, why is it epigenetically stable? So what we've got is our protein complex. And of course, when we talk about this recruitment in molecular biology, we sort of draw one protein, recruited to the site in the locus, and we're done. That's the schematics, and they stay stably associated. But of course, as people are doing live imaging at the moment, what's clear is that transcription factors rarely stay very long at the locus. So what we've got, I can get this to work, is that actually it goes, but then something keeps it there. So it's not what takes it there, it's what keeps it there. So now I'm going to go into our understanding of what on earth keeps it there. And this took us back into a part of this VIN3 protein. This is this cold-induced thing I was telling you about, but looking specifically just at the C-terminal region. And what I'm going to show you is this is actually a little polymerization domain, and this is work we did with Marianne Bientz at LMB. Mark Fiedler was the first author. And you've got to think of this little domain now as a molecular Velcro. It's the best way of thinking about it. When we did the structural analysis, these 4 helices that make up this Vel domain, as we called it, it's like a Lego block, a single Lego block. There's a head and a tail end, and they fit together in polymers. And I haven't got this on here, but if you put it under the microscope, it forms these huge long fibers of head-to-tail polymers, where just the protein is there. Now, when we then looked at very carefully, we made mutations, 1 amino acid change in the head and 1 amino acid change in the tail, was sufficient to completely block this polymerization, and that made the plants completely blind to cold. So that polymerization is absolutely key for this epigenetic silencing. Now, this is now current work, so we have here model, because we haven't really proven this. So what do we think is happening? So Marianne, actually is sort of the world expert in polymerization domains. I whizzed through past it, but there are very few characterized in biology because they're so hard to get any idea of the structure. Because they tend to polymerize in vitro, which means they all fall out, they're insoluble, they fall out of solution, they're absolute nightmare to work with. So you have to sort of guess the interface, mutate the interface before you can actually make them soluble and then get the crystal structure. So you have to go around a lot of circles, and most people don't bother. But the view is that actually what they do is they basically act like a Velcro. They increase the local concentration of the protein that they're associated with. And therefore, any ligand associated on the rest of the protein is then at high concentration locally in one place. Now, if you look, so this is the schematic then, what we would have is this is our nucleation region with our polycomb. What you've got is multiple VIN3s then. So you have this Velcro with numbers is the importance here. The affinity is not high, but if you have plenty of them, the avidity is high enough to then get you over this nucleation threshold and give you sufficient numbers that when you divide during replication, enough can go to each daughter strand to recreate with the molecular Velcro feedbacks, the epigenetic complex is then transferred. Now what then, so this is the Vel domain that gives you that local concentration. What is this binding? And that's, that is the question of the day, certainly in my lab. And in the polycomb field is, what gives you the specificity for silencing in the chromatin world. Now, it's this relatively small 3-nucleosome region, and if you look at what these other ligands are on these proteins, well, we have a PhD domain. We have all sorts of domains that smack of chromatin association. And actually, we looked at that again in Marianne's lab. But the PhD domain that we've got in these proteins actually has a very odd one. Didn't look at all like the others that have been characterized which bind histone tails. And in fact, when we've got the structure, it's totally the wrong charge. We've got this very positive charge, which is really not going to bind to positively charged histone tails. And when we've done a lot of biochemistry, it does not bind histone tails. So we don't really know what it does bind at the moment, but it's something probably quite negative, maybe a different aspect of the histone, maybe something to do with the DNA. It's not strongly binding DNA. It's certainly not sequence specific. So what we are doing now is actually we have a very exciting collaboration with Rosana Colipado-Guavo's group with Jan and Maria, who I happily see in the audience here, where we're doing the nucleosome modeling. Remember, this is only 3 nucleosomes. And what these guys are doing now is to model the nucleosomes and to understand what's going on in the different temperatures and how these proteins then influence that nucleosome structure so that we can then make some predictions and test, is there a specific kind of structure in combination with perhaps the PhDs talking to a specific aspect of the histones and maybe a bit of the DNA as well, but to really understand what drives polycomb specificity. And I think this is a great system to actually really understand that for the first time. Okay, so I've told you about this switch, which involves this stable association of this protein complex at this specific site in the gene. And this is then giving us some kind of registration or counting mechanism. And you'll have seen that the cells were switching sort of slowly, happening stochastic switching over time. And that gives rather a nice counting mechanism. It's sort of like each allele is given a vote. Have I had enough cold? When it votes, it's nucleated and then remained stably nucleated. And it takes a while for the whole population to switch. So that, in effect, answers that question, how are plants registering the prior exposure? Now, again, I think this is not plant-specific. None of these are plant-specific. So therefore, this is an interesting concept that might be more broadly relevant. So let's think about what's that second question. So how is the noisy temperature information judged? And I just want you to reflect that this sort of schematic here shows that this temperature profile is happening and the downregulation of the gene much faster than the accumulation of the accessory protein. So the shutdown of transcription, which is absolutely required for the polycomb switch, is much faster than the actual switching mechanism. So different timescales are operating here. So now we're just going to look into what is the plant registering. And that took us into this co-transcriptional word. I'm sorry for the title. It's a word that people kind of think, well, what does she mean? It basically means something that's happening during transcription of the gene. And I'm not going to get into too much details, except all our avenues, as I said, coming back to FLC, but not only that, kept coming back to antisense transcripts at FLC. So if this is the structure of the gene I showed you before, and this is the poly-A site of the sense transcript, there's a promoter here, where you get a series of antisense transcripts which are alternatively spliced and alternatively polyadenylated and terminated. And we just keep coming back to those. Now, they've been a bit controversial. What do we know about them? Well, we know that actually, if this is the schematic of the time frame of the VIN3 induction, then the antisense are cold-induced, and there's a pulse of antisense transcription associated with shutdown of the sense transcript. And because it was a switch that we were dealing with, we wanted to look not just at the sort of RNA levels, but actually at each allele. We ended up doing some FISH to actually look at individual nuclei using probes. So anything red here is looking at FLC sense transcripts, and anything green is then the antisense. And these are individual nuclei. And what you can see is you either see green or you see red in the different nuclei. I'll show you some more pictures later. But this was what came out, is that we only saw mutually exclusive expression of sense or antisense at each locus. Well, you could imagine that made sense, because if you have a pulse of antisense, that's going to help shut off the gene. So that was made sense. But the other thing was that these green were always bigger foci than the red. So we've sort of termed them now these clouds, because they look just like the exist RNA over the inactive X in mammalian cells, somehow involved in silencing that inactive X. And just to show you a few more pictures, they're not only present in the cold. You see in a few nuclei, about 3% in the warm, you do see them, but they seem to be cell cycle regulated, the expression of the antisense in the warm. And then what happens, as the plants start to sense the cold, many more cells, nuclei, switch the antisense on. But there was always sort of two big foci, which then, this is 1 week, 2 weeks, 3 weeks exposure, sort of dissipated, sort of fitting with his big pulse and then dissipating. And through some amazing work, and this was not easy in plants, Stephanie Rosa, who did this work, then did the DNA fish to ask, are these really big puncta actually over the gene itself? And yes, indeed they are. So it's just like exist. You have this pulse of transcription on the antisense, and then the RNA hangs around the locus, somehow involved with the silencing. Now, we've done, and I spare you a lot of detail here, but you'll see in the literature lots of knockouts and overexpression through the genetics and all sorts of things. But there's still a lot of controversy out there, just as there is for lots of non-coding RNA. And in fact, non-coding RNAs, some people love them and some people hate them. It was a bit like the Mark Potashny experience, sort of. But what's, I think, emerging is that they don't give phenotypes like protein knockouts. They affect the dynamics of the process, and therefore you don't often get strong phenotypes. But we kept coming back to finding them through different routes. And I just want to give you a couple of examples of that, because it then said to us these were important. Even though when you do a knockout, potentially the phenotype's not that strong, you have to look in the right place. So how does it regulate? Well, through multiple mechanisms. And each one of these different forms seem to regulate the gene differently. Okay, so I'm going to tell you a little bit about this first one. And again, totally by accident, we bump into it. This accident happened to be field experiments, because we wanted to understand the adaptation of Arabidopsis thaliana to these very big, different geographical range it adapts. And the ERC, I always thank them funded this. I'd never done a field experiment in my life, okay? But we said, we're going to do field experiments in Sweden, and we're going to understand, what the silencing looks like in a real winter. And then, so we had some field experiments in North Sweden and some South Sweden. We hooked up with people who had universities nearby, and some fantastic master's students helped us. We planted them all in September, lots of genotypes, non-transgenic, obviously, in Sweden. We had a mix of a cannibalized greenhouse in Norwich so that we could do some transgenic ones as well, but there was no heat and no light. And then we harvested every week over a winter period to say, what's going on. This was one of those experiments. You see here's the temperature profile. This happens to be a South Sweden field. And this happens to be we looked at lots of things. There's a lot of work been published already. But there was one day suddenly, these antisense transcripts, which I forgot to say we christened couleur, actually, because of the hot air for the mammalian polycomb. Anyway, couleur suddenly shot up on one day. And it's like, well, okay, what's going on that day? And that day, when you looked at the temperature profile, blown up a bit here, was the first night the temperature dipped just below freezing. So we thought, gosh, that's intriguing. What's going on? And so we went back to our chambers and recreated the same thing in our chambers where everything else was the same. So we either gave the plants 5 degrees constantly over our 24-hour period, or fluctuating over a mild fluctuation, or a strong fluctuation. And it was only the strong fluctuations that dipped below 0. But you remember that sun clock model? That would predict these would all give exactly the same effect if it was averaging over 24 hours. What we found was they gave very different effects. So couleur was induced by the constant cold, which is what we'd always done. The fluctuating mild wasn't so different, but the fluctuating strong conditions, we saw this superactivation, just like we had seen in the field. And that was associated with super repression of the sense transcript. Now again, I've missed out a lot of details. We've begun to unpick what on earth's going on. It's something to do with the antisense invading the DNA, forming an R loop here, and then requiring to be processed by transcription termination. And this is now what we're working with Laurie Passmore at LNB. And that proximal termination event creates and changes the chromatin environment over the gene. Happy to go into this if anyone wants to talk about it later. But what for this talk I want you to focus on is that this particular mechanism is very good at monitoring those freezing nights. And remember, you're a plant. You're trying to work out, is winter progressing from this complicated temperature profile? So the first few freezing nights, that's a pretty good clue that winter's coming. Is that the only way the antisense work? Well, no. Again, by accident completely, we run into this second isoform. And that turns out to be important, involved in a protein that activates FLC. So this is how you make high levels upon germination, okay? This is a protein called Frigida, which we cloned many, many years ago. It interacts with very conserved transcription factors, mediator, all of the goodies that most promoters have to give high-level expression, and our green-colored blob active modifications. And if you look at the GFP, you see it's all diffuse in the warm-grown plants. But what we realized was that after a couple of weeks, this Frigida GFP, now you see these punctate shapes coming. And this is just a schematic, really, of what's published in this paper, is that what we find is that it's a very strange protein. It's intrinsically disordered and has two coil domains, Frigida, without much else. But it associates specifically with this particular Coulet isoform to then be sequestered into these foci, which do not co-localize with the gene. So it's a way of ripping off the activator, off the promoter during the cold. Now then we want to say, well, what's the dynamics of that? How fast is this operating? And we realized that we could take these plants, so we're looking at root cells here, and we grew them on the microscope and then just change the temperature. So at the top, you're looking at now plants that were warm and then cooled, and then slowly, this is hours. You'll see a sort of 5-6 hours, now the puncta have appeared. And now if you look at the bottom, and that fits with the decline of FLC expression. So this is another mechanism for shutting the gene down. And then when you cool it down, you see within 4 hours now, you warm it up after you've cooled, they've all dissolved again. So over a sort of 3 or 4 hour period, you basically have this activator sequestered or delivered back to the gene to graded in a temperature-dependent way the transcriptional input into FLC. So what the plant is doing is looking at this complex temperature profile and looking at different aspects. So when it's nice and warm, you have a diffuse Frigida. You allow it to activate the promoter. But even within 3 or 4 hours, you then, if it starts to cool down, you sequester it away. So that's one mechanism. But there's a different one too, involving the antisense, a different form of antisense, in that every time you start to get cold nights, now you have this other way of shutting off FLC transcription. In addition, you of course have the accessory protein accumulating by reduced dilution through the reduced cell division. But any time, and that I showed you a while ago, when you had these afternoon spikes, all of the Vin3 that's accumulated slowly, if you have an afternoon spike, you turn all of that Vin3 over. So when you look at this, you think, gosh, this is very sensible. If you were a plant trying to judge that temperature profile, you just have a few key nodes in here that really help you work out, is winter progressed? Is it nearly time for spring? So I'm coming to near the end. I just want to say we wanted to look at these two questions First of all, how you read the noisy temperature profile, and that's taken us into looking at these co-transcriptional mechanisms involving the antisense, but also transcriptional effects of FLC. And then that's happening in a graded manner. And then you have the registration, the switching, to actually give you that counting mechanism so that you can register prior exposure. So those two questions have led us down to interesting, fairly mechanistic route, but I think they do potentially relate generically. And I just want to finish on a couple of points. One is that going back to this sequence of events that was always the sort of way we do environmental experiments. There's a signal, you take the signal transaction and you look at what's happening at the end. This just shows schematically that we can actually divide up these two phases, this transcriptional downregulation and this epigenetic silencing, into mechanistically quite separate events. So the transcriptional shutdown is graded. This shows a schematic now of a chromosome with our locus. This is the FLC locus, diploid. So with increasing cold, this graded shuts off. But then, quite mechanistically separately, you can have a switch to an off state. And once you're off, you then absolutely stay off. So those are mechanistically independent and can flex with different temperatures independently of each other. Now, it's always hard to understand. So the metaphor for here is if you're in a car and you're driving up to a roundabout and you've got plenty of time, well, you take your foot off the brake and you kind of cruise to the roundabout just at the right speed. But suddenly if something happens and you need to put your foot on the brake, well, then you can actually shut down much faster. And so it's these two mechanisms to shut you down with the right timing, given the exposure that you're experiencing, that is what I'm trying to show here. And this basically says that, we really shouldn't think about exposure this way. Environmental exposure is done, there's multiple inputs, and you have these different integrated, interconnected effects which can be working mechanistically parallel. So this gets awfully complicated, and I think Escher's hands is a wonderful analogy for that. But it sort of says, beware of doing experiments with lots of omicky type things and expecting a sequence of events, because I think for complex signals that's not going to happen. And that just leaves me to thank lots of people. This was a picture actually when the Wolf Prize was given. It was in the middle of COVID, so we had to take a picture and everyone had to stand. We all had to get dressed up in black tie on a Monday morning while they took a picture. So I just thought there's lots of people who've left already. Of course, I've given an overview of lots of work. The two I want to point out are Anna Schulten, who's still in the lab, and Miguel Montes, who actually has arrived since this picture, who are the two people. He's working on this project with the PRE, the accessory, what takes polycomb to the certain sites, and Anna's doing the work on the avidity. And we've had some amazing collaborations, some still ongoing, very exciting, and some fantastic funding that's made all this possible. And thank you for your attention.
Thank you very much, Caroline. That was fabulous. Time for a couple of quick questions. We have to do a bit of plaque unveiling business.
The students I had lunch with promised me they were going to think about questions.
Look at this. And our students fabulous. There's one up there.
There's got to be some.
So there's also cell-to-cell noise, so as well as temperature noise. So does the system cope with that noise in a way that just always reaches the same end, or do you think there are mechanisms where cells have to talk to each other to even out things as well?
Let me see. Well, what I showed you, those routes where you could see the lineages, the cells next door were acting completely independently. So there doesn't seem to be any communication from one cell to the next based on that expression profiling that we can tell.
Thanks, Caroline. I think the VIN3 protein is quite interesting. And I was wondering if you know if it has other targets.
Yes.
And also is it always with the PRC2 complex, or is it on
Yes.
chromatin and then the PRC2 complex comes and goes? Which,
Yes.
how does that work, or do you know?
There's probably 1,000 targets in the red box's genome. We've published the ChIP-seq, so there's lots of data out there for that. As far as we can tell, when we IP PRC2, we don't always, well, you have to do that out of the cold-treated plants, of course, to actually see it, because the expression's only in the cold. When we IP Vin3, we don't get sort of stoichiometric polycomb, but when we look at the targets and where the polycomb is, they coincide quite well. Now, I wouldn't say, I could put my hand on my heart and say always, but there's a good correspondence. So I see them very much like the mammalian PRC2 accessory proteins. They're there to nucleate, and I didn't really get into, other things happen downstream of that. They're not needed for the downstream part. For the long-term memory, they're needed for that initial switch.
Hey, sorry if I missed it, but do you know exactly what's regulating kind of the slowing down of growth in response to the cold, which actually lets the Vin3 accumulate?
No, you didn't miss that. But temperature sort of affects everything. So, biochemistry slows down in the cold. Whether or not, we don't really know. No one's really actually elaborated that. No one's actually asked the question. But, cold affects everything. Temperature sensing is distributed all the way through the network because temperature affects everything. So my guess would be, it just slows down because everything slows down. But, yeah, it's a good question. We don't know the exact answer.
Richard?
You've got FLC being turned off in this sort of cell-autonomous way. Then the decision whether to flower or not is an organism-wide decision, surely. So it has to go the other way.
Yes. So do you remember I said that it's averaged out because the target of FLC is FT gene. And FT protein moves around the plant. It's a very small protein. So basically all of this mosaic kind of silencing is then averaged out by the FT moving around the plant, getting to the apex, the shoot apex, which is, of course, where the flowering occurs. So it's then a graded signal getting to the apex from all of this salt and pepper around the plant.
Billy?
In this model here, the epigenetic decision is allelic specific.
Yeah.
But in the co-air expression, it's not, or at least it's not portrayed like that. Any chance that there is some allelic difference in the co-air expression that then will allow the epigenetic?
Yeah, I think there's probably a lot to that. I don't think we've had the tools to exactly understand that yet. We did many years ago actually a LACO tagging where you can see quite quickly, and just like the X chromosomes, they come together transiently. And that's a very important part of determining which X gets switched off. So there's more to it, I think. Yeah. Although this has just come out, this is quite important to me in that when we do the freezing, we really speed up that top pathway. That's what happens in the sort of FS condition. We don't change the bottom. So now actually you can change the relative effect on accumulation of the histones from the transcription. So you can see them flexing separately, but you can also see how advantageous that would be for the plant to cope with very different winters. Autumn's really very different each year, and the plant somehow got to adapt to that, but not switch off its FLC too fast. So I think that's an important part of this.
There you go.
So from what you know, that was a lovely talk, thank you. From what you know so far, what do you think our 15-degree afternoons in February are going to be doing to our crop plants?
Well, your Vin3 is all going to be turned off very quickly. It's making a very big difference to the crop plants. I mean, the brassicas particularly. The very strange kind of very cold just before Christmas a little while ago. This is why the supermarkets suddenly, you don't have any broccoli for a month, because it's just all either come all at once or it's been disrupted because it's not vernalized properly. But for brassicas, this whole temperature dependent thing is hugely important for scheduling a nice, secure market delivery. And the supermarkets are very worried about this.
If we have no more questions, it remains me to thank Caroline once again for a fabulous talk. And a very worthy contribution to Saunders series. So now we have some bling, which we're going to adorn the department with. So I'm going to invite Professor Ferguson Smith, the current Arthur Balfour Professor of Genetics and President of the Genetics Society, up to assist me in this. So in 2019, the Genetics Society had its centenary and had a wee celebration. There was a nice party, particularly enjoyed the wine and drinking, and as part of that, Sir Paul Nurse unveiled these two plaques to celebrate and honour the founders of the Genetics Society, Edith, Becky Saunders and William Bateson, who founded, as it was at the time, the Genetical Society. Actually, they were made of polystyrene, but these are shiny ones that we will use to adorn. The Genetics Society decided it would probably be best if we were willing to put them in a new location, and that would be the Department of Genetics here in Cambridge, which of course is the first and best Department of Genetics in the world. Founded.
The only one.
No. Oh, no, it's the only one left in the UK. Founded in 1912 with the appointment of Reginald Punnett as the Arthur Balfour Professor of Genetics, and as I said, that is the first permanent chair of genetics established. I would say that the department at that time was a room at the back of the professor's house, and however, it has gone on from strength to strength since its formation in 1912. Now, when we were doing this, I was getting a bit sick of other departments in Cambridge putting up bling about their Nobel Prizes, and in fact, in the Department of Genetics, from 1978 to I think about 1990, when he went to the Gurdon Institute, Martin Evans was a faculty member in the department, and in 1980 he began work with Matthew Kaufman that led to the development of mouse ES cells, and then afterwards, with very important work involving Alan Bradley, who I'm happy to see is in the audience here, and Liz Robertson, actually, generating germline chimeras. And this was recognised, Martin's contribution here, was recognised by the award of the Nobel Prize in Physiology or Medicine in 2007, along with Mario Capecci and Oliver Smithies. So as I said, I was tired of other departments putting up their aged stuff. So the work that led to this Nobel Prize was carried out in the basement of the genetics department. So I'll invite the president of the Genetics Society and Arthur Balfour, Professor of Genetics, to unveil them, as it were, metaphorically.
Well, I'm not unveiling them, but it's, the Genetics Society, on behalf of the Genetics Society, I'd like to say how proud we are to be partnering with the genetics department to really celebrate the birth of genetics in Cambridge, and the contribution that Becky Saunders, William Bateson, and actually belatedly Martin Evans, very belatedly those two, but also belatedly Martin Evans has given to genetics in general and to our community too. So I think it really is wonderful to recognise what they've done. It's wonderful that we were able to persuade the Genetics Society that these belong here on our buildings on the Downing site, and to really value the work that they've done. And it's particularly fitting to be doing that on this day when our Saunders lecture has been given so beautifully by such a wonderful plant geneticist as we celebrate Becky Saunders. So it's a really, we're not quite sure what day we're going to put them on. And
we'll be nailing them up when we can get someone from the estates division to come in.
So that'll be another
so
5
in 2
years.
years' time, we'll be able to show you photographs of them actually on the wall.
So Becky Saunders was the 4th president of the Genetics Society, and it's really great that the 3 of them are going to be recognised in this way. So.
First meeting of the Genetical Society had 32 attendees, mostly from the John Innes Centre here in Cambridge.
In fact, Arthur Balfour was the first president of the Genetics Society, apparently. I didn't know that. Anyway, so many congratulations to these 3 wonderful people,
yes.
Whom we owe a lot. And I guess we're going to invite you all back to the genetics department to have some more celebrations.
Yes, and there's an extensive poster display on the life and works of Edith Rebecca Saunders, which was developed when we started, when Anne put this whole process in place about these Saunders lectures by our former librarian, Christine Alexander, who's developed some fabulous work, and really you can go and read about Becky's contribution to genetics, to plant sciences, and to the betterment of and training of women in science. So thank you all once again. Thank you especially to some of our alumni who've come along. It's a pleasure to have you here, and we invite you back to the department for a wee swally, as we say in Glasgow. Thank you.
Thank you.